All right, so the last section of chapter 13 deals with the largest of our sugar types, which are going to be our polysaccharides. And what your polysaccharides are is they are polymers of monosaccharides. Okay, and what a polymer is just it's a huge molecule that has many of these building blocks or building units. And the monosaccharides that we're going to put together to make these polysaccharides are going to be glucose. So all of the polysaccharides you're going to be looking at are going to be polymers of alpha D glucose. So these are large molecules made by combining many monomers. Right, what a monomer is just a building block of a polymer. And the monomer that we're going to be looking at here is going to be one that we've seen a lot of previously, our alpha D glucose. So the first couple polysaccharides we'll take a look at are amylose and amylopectin. And the difference between them really has to do with how many branches or the degree of branching that we find in their structures, right? They're all going to be made up of many monomers of these alpha D glucose sugars. It just depends on how they are arranged. So if we take a look at amylose, right? Amylose ends up being a straight chain, and this is what we see here. Right, so there's your amylose, and all it is just a bunch of these alpha 1,4 glycosidic bonds, which we saw previously, that connect one glucose molecule to another one, to another one, to another one, etc. on both sides of this, right? So your amylose is a straight chain, or it's just an unbranched chain, I'm sorry, of, hundreds, thousands, millions of these glucose units. And the amylose itself, even though it's a straight chain, right, it actually coils. So it's an unbranched chain. It coils up, right, and it actually looks like sort of like a spiral. So each of these little colored hexagons here represents an individual glucose molecule in the polymer of amylose, right? So it's an unbranched chain. It just sort of winds around itself like a spiral staircase. In the case of amylopectin, right, amylopectin is still made up of glucose, right? So you have chains of glucose that occasionally branch out. Right, so this is a chain of glucoses going from left to right, for instance, and what the bond that holds those glucoses together will be the alpha 1,4 glycosidic bond right here. And occasionally you're going to have these branches that branch off of the chain, and those branches connect another chain with the first chain by an alpha 1,6 glycosidic bond, right? And it's alpha 1,6 because we're connecting carbon 1 to carbon 6 of the other, and it's alpha because all that your polymer is made up of are these alpha D glucoses, right? So the alpha 1,6 glycosidic bond, that's the branch that branches out, and then there's another chain of the glucoses going from left to right, and then that one may branch out in another case as well. All right, so the difference between amylose and amylopectin has to do with the degree of branching. In amylose, there is no branching. In amylopectin, there is branching that occurs. So amylose, both amylose and amylopectin make up starch. Amylose makes up about 20%, about 1/5 of a starch sample. And like I said, it's made up of these alpha-1,4 glycosidic bonds that connect our glucose molecules. It's a chain, even though it's coiled into a helix or a sort of like a spiral staircase. Like a coil or a spring. Or a spiral staircase. Amylopectin makes up the remainder, about maybe 80% of starch. It is branched, and those branches are due to the 1,4 glycosidic bonds. What keeps the chain together are the regular 1,4 glycosidic bonds. Starches themselves can be broken down, so they hydrolyze. So they can be broken down to give smaller saccharides, or smaller sugars that we call dextrins, that can then be broken down even further to give even smaller things like maltose, and then finally glucose. Right? Glucose is the end product of all polysaccharide digestion, because glucose is sort of what your body uses for energy eventually. Glycogen is the third polysaccharide we'll take a look at, and glycogen is also a polymer of glucose, and it's stored in the body. It's stored in the liver and in the muscle of animals. In terms of its structure, it's very similar to amylopectin, but it's more highly branched. All right, but it's the same type of structure that we'll see, meaning that we'll see those 1,4 alpha-1,4 glycosidic bonds that keep the chain together, and then occasionally, but happening actually more frequently now, we're going to see the alpha-1,6 glycosidic bonds that are due to the branches, that they're going to occur more frequently than we would see in amylopectin. All right, so we can say glycogen structurally similar to amylopectin, but more branched. Right, so glycogen is the way that we store glucose in our bodies. We don't store starch. We don't store glucose in the form of starch like plants do. We store glucose in the form of glycogen. So oftentimes you'll sometimes you will hear glycogen being referred to as animal starch. Right? It's the way that we store glucose in our bodies. Cellulose is the last polysaccharide that we'll take a look at. Cellulose is found very commonly in wood and also in the cell walls of plant cells. Okay, regular human cells don't have cell walls. Okay, and we don't have cellulose in our cells. The plant cells, though, have a layer around their cell membranes that sort of keep the cell intact, and that cell wall is made up of cellulose. It's the major structural unit of wood. It is also a polysaccharide of glucose. It is unbranched. One of the main differences, though, between cellulose and all of the other polysaccharides is the type of bond that links those glucoses together. And in the case of cellulose, that bond is a beta 1,4 glycosidic bond. All right, that's a huge difference between cellulose and the other types of polysaccharides. So contrast to the other. It may not seem like a big deal, right, having an alpha versus a beta 1,4-glycosidic bond. But it is a huge deal in terms of what the organism is able to do with that bond. So your cellulose, just some general properties of it. It doesn't form hydrogen bonds with water, so it tends to be insoluble in H2O because of the way that the cellulose is actually packed. It gives a very rigid structure to the cell walls, but also in wood and fibrous tissues in plants. It is more resistant to breakdown or hydrolysis than starches are. And the key thing here, the very last bullet point, is that it cannot be digested by humans because we don't have the machinery or the mechanism to break that beta 1,4-glycosidic bond, right? Even though we can break down starches, for instance, which have the alpha 1,4-glycosidic bond, the mechanism in our bodies, the tools that we use, like our enzymes, they do not recognize that beta 1,4-glycosidic bond in cellulose. So we cannot, for instance, go out, eat some grass, and get any energy from that, right? It doesn't work that way, right? As you know, typically the fibrous tissues of plants, for instance, they normally just go right through us. It's fiber. We don't get any caloric value from that because we cannot break down or digest that beta 1,4-glycosidic bond. So let's take a look at what that looks like. And we'll see the slight but very important difference between this and the alpha 1,4-glycosidic bond. So let's just focus in on a couple of these. So let's just take a look at maybe these two here. All right, so we have one glucose on this side and one glucose on the other side, and there's the glycosidic bond between them. All right, so again, it's connecting carbons 1 to carbon 4, so it's a 1,4-glycosidic bond. But if you notice closely now on carbon 1, so if you take a look at the OH that was connected to carbon 1, right here you can see that OH is actually pointing upwards that way, and that makes it the beta isomer, right? And that's the case in all of these. The carbon 1 in all of these had that OH pointing up. They were all pointing up, all pointing up. And that makes them the beta form of this bond, the beta 1,4-glycosidic bond. And that's a huge difference between cellulose and all of the other polysaccharides that we talked about, where we saw the alpha 1,4-glycosidic bond. All right, so cellulose then, still it's made up of glucoses, but the bond that connects the glucose is very different than the other polysaccharides.